So I'm a hematologist and oncologist, and I'm also a microbiome researcher at Stanford. I'm delighted to be here today in sunny Chicago. It's a little cooler than where I live, but absolutely beautiful to see the fall colors. And I'll talk to you today about a huge wave of data that is coming at us. You've heard from John, you've heard from Jack about reams and reams of microbiome sequencing data that's being generated, being used to define changes in ecological systems, being used to define risk factors for surgical outcomes that are poor. And my particular lens is a little bit different as an oncologist. So the outline of my talk, I'm going to talk about some bad diseases and a risky treatment. I'll talk about a common phenomenon. I'll then touch on one simple question, two hard problems, a new technology, and finally, I hope to convince you that we have some intriguing results. So with that, let me introduce our bad diseases and our risky treatment. So we study patients who have malignant hematological disorders in our laboratory. Put another way, we study people with white blood cell cancers, red blood cell cancers. Okay, so these are people with leukemia and lymphoma. So just by show of hands, because I know it's a little bit late in the day, how many of you know somebody who's had leukemia or lymphoma? Raise your hand. If you look around the room, you'll actually see that about a third to half of us have raised our hands. So while we consider these diseases relatively rare, they're not quite as rare as we think they are. Okay, so those of you who know people who've had leukemia or lymphoma, how many of you know somebody who's undergone a bone marrow transplantation, also known as a stem cell transplantation? Okay. So a bit fewer, maybe about 50 to 75% of those of you who raised your hands before. And so while this is also considered a relatively esoteric treatment, it's not so uncommon as we think. So let me describe what this therapy is for those of you who aren't physicians in the room. The idea is very simple. We have a recipient. The recipient is the person who has cancer. In order to prepare them for their stem cell transplant and also to decrease the bulk of cancer within their body, we treat them with very high doses of anti-cancer drugs. Okay, this destroys every rapidly dividing compartment within their bodies. Hair falls out. They have denuding of their gut, and they also lose their hematopoietic stem cell compartment. We often also will treat with radiation therapy at this time to achieve the same objective. Okay, now, of course, not having any red blood cells, white blood cells, or platelets is incompatible with life, and so we have to rescue them. And the way we typically choose to rescue people who have malignant hematological disorders that are aggressive is with healthy hematopoietic stem cells from a donor. Classically, this used to be collected from the bone marrow in adults. The biggest area or niche that has hematopoietic stem cells is in the hip. So we would take a big needle, stick it in the hip repeatedly, pull out bone marrow, isolate CD34 positive stem cells from this population, and then administer them to the individual who had been previously irradiated. Nowadays, you guys probably know we collect stem cells from peripheral blood and umbilical cord blood as well. And the idea is you inject those healthy hematopoietic stem cells, and then you just sit around and wait. Okay? And you sit around and wait while all kinds of terrible things happen. Okay, so on this rough timeline that I've outlined up above is on the far left, the point at which we initiate the bone marrow transplantation. Right, so we give the chemotherapy, the radiation. Just like John talked about bowel prep, we have this very low evidence practice that we often use, which is called gut decontamination, which is making the assumption that every bacterium within their body is terrible, and so we will eradicate them from the face of their body, and we give broad spectrum antibiotics. Of course, this is complicated by a lot of organ toxicity. At the dotted line, we introduce the stem cells, and like I said, we sit around and wait. During this time that we're sitting around and waiting for a competent immune system to be rebuilt, patients are set up for infections, relapse of their original cancer, graft versus host disease. Remember, we've just created a human chimera, so the immune cells are actually recognizing the host as foreign. There are lots of idiopathic disorders that we have no idea the cause of. And of course, if patients are lucky enough to make it out to a very late time point, they often get secondary malignancies. And these secondary malignancies are often due to either the DNA damaging agents that they received in the upfront setting or due to infections that can cause malignancy. And so this is my fundamental problem with what we do. Our therapies provide a chance of life for people who otherwise would have succumbed to their illness, but they do so at a very high cost. About 30% of our patients die in the first year after an allogeneic stem cell transplant. of those 30% of patients who die in that first year, only about a third of those patients, 37%, die of relapse of their cancer, which means that about 2/3 of the time, as an oncologist, I have beat their cancer, but I have most definitely lost the war because patients succumb to other disorders like graft versus host disease, infection, and other. And so mortality due to infectious complications in this patient population is unacceptably high. And we've done very little to actually move the needle on this, with the vast majority of folks in our field focusing on the cancer relapse aspect of this pie chart. So next, I'll bring you to a common phenomenon that we see in our patients, and I'll try to convince you that maybe it has some clinical relevance. So this is leveraging a stool biospecimen bank that we have at Stanford Hospital that our laboratory built. We have about 1,200 samples from over 500 patients. We actually have just initiated a process to sequence them all. And what I have here represented is a stacked bar histogram. So each color represents a different genus of bacteria. And this is a time course. So over on the far left is a relatively normal gut microbiome. You can see there's some variety of colors. And this is a bone marrow transplant patient who was seen at Stanford Hospital. Day -8 is the day before they receive any chemotherapy or antibiotics. Day -2 is just before they receive the stem cells. By convention, we call the day that they receive their stem cells day 0. So you can see already between the time of the start of this experiment, this experimental therapy, and day -2, when they've gotten exposed to chemotherapy, antibiotics, the microbiome has become very undiverse. This patient then went on at day 7 to develop fever and neutropenia. And when patients have fevers and they have no neutrophils, they are a setup for infection, obviously. So even if we don't find an offending bacterial cause of disease, we treat them with empiric antibiotics. It's the kind of better safe than sorry sort of approach. And you can see at that point where we introduce antibiotics for the empiric treatment of FNN, now we see these green organisms popping up. This patient then got graft versus host disease. Remember, the immune system from the donor is now attacking host tissue. This disease also has been shown to be linked or associated to the microbiome. We treat this disease empirically with steroids to dampen the immune system. And now you see these purple organisms popping up. The diarrhea eventually resolves. And this is what happens to this individual's gut microbiome over the subsequent about total of 100 days. And one thing you'll notice is that there is dramatic gut microbial diversity loss post-transplant. And many of the diseases that have been associated with microbiome changes have been associated with this same thing, low diversity, bad outcomes. Okay, so this is just another example of low diversity and a bad outcome. This has been termed oligodomination, when you have one or a few organisms dominating an otherwise expectedly diverse niche. And the question we had was, well, like, so what? I mean, we see this is a phenomenon, but does it have any clinical relevance? You already heard about a disease today that does occur as a consequence of oligodomination in many cases, and has major consequences. This is C. diff. So this is a picture of pseudomembranous colitis, an extreme version of C. diff colitis. And as you know, many of us walking around have C. diff within our colons and are doing just fine. It's only in the setting of loss of diversity of the gut microbiome that people actually end up getting clinical disease. Okay, so the risk of oligodomination includes opportunistic infections. It is also associated with bloodstream infections in this patient population. You know, more broad spectrum studies or broad scale studies have evaluated if low diversity is specifically prognostic of bad outcomes in bone marrow transplant patients. This is a study that was done by Eric Paymer's group at Memorial Sloan Kettering. In this study, they collected stool samples from patients undergoing bone marrow transplantation at the time of engraftment, so about 30 days after their transplantation. They then did 16S sequencing to characterize the microbiome, and they divided the patients into 3 groups. Those with low diversity, medium diversity, and high diversity. High diversity is the blue line, low diversity is the red line. They then looked at these patients' survival over time. This is just a standard Kaplan-Meier curve, and what you can see is that there is a statistically significant difference between the high and low diversity groups. In a multivariate analysis, it was actually shown that low diversity of the gut microbiome was one of the best prognostic indicators of poor outcome, period. Okay, more than underlying disease, comorbidity index, et cetera. So oligodomination happens, and it seems to have some clinical relevance for us. So this actually was followed by one simple question, which is, how does this occur? And this was a question that was posed by two very talented graduate students in my lab. Eli Moss, pictured on the left, is a genetics graduate student. Alex Bishara on the right was a computer science graduate student, now has stayed on as a postdoc. And interestingly, as a physician, when I saw oligodomination, I was kind of like, well, we see this, guys. And they were like, no, but why does it happen? And I was like, no, we see this. End of conversation. And I'm actually glad that they pushed, because they said, well, I don't understand why these same organisms win. We've actually now, I mentioned, sequenced a majority of our stool bank, and we actually see oligodomination in over 20% of our patients. And we tend to see typical organisms popping up. So here's another individual. This is an individual who underwent bone marrow transplantation between time points A and B. And again, this patient gets dominated by, in this case, the light blue block, which is Bacteroides cacchi. And so how, at the genetic level, they asked, does this oligodomination occur? Is there something about the genome of these organisms that dominate, that predicts whether or not they succeed in this difficult environment? Let's talk a little bit about what this difficult environment is. And so this is this same patient, patients who we just saw previously, time points A through B. Remember, between A and B, at that red line, the patient underwent their stem cell transplantation. You can see that the patient had lots of complications, as one does. You can see they got exposed to lots of antimicrobials, so antibiotics, antivirals, antifungals, immunosuppressive agents, chemotherapeutics. You may remember from that previous slide that between B and C was when you saw that big jump in kind of oligodomination. And you can see that between time points B and C, the patient was exposed to tons of antibiotics. Okay, so what do you think happened? Why do you think this organism won? Maybe it's got lots of antibiotic resistance features, right? So maybe this organism is particularly good at resisting antibiotics. Interestingly, if we were just to sequence all of the DNA from the stool and look for antibiotic resistance genes, we don't actually find a statistical enrichment of antibiotic resistance genes in these organisms. So it doesn't seem that they just have more of these genes. It could be that they're acquiring some sort of resistance, but how would we measure that? So, in this particular case, just to remind you, the B khaki bloom corresponded to broad-spectrum antibiotic exposure. And so we're going to ask now, what is it about this organism that actually caused it to win? And remember, this is a pretty generalizable phenomenon. So here we ran into a problem. In fact, we ran into two hard problems. When I look at all of you and I think about your gut microbiomes, which I assure you I did earlier today, I look at your gut and your stool as an exploration opportunity that is akin to going to Mars. Like, I guarantee you, if I sequenced everything in your microbiome, I would discover new organisms in each and every one of you. In fact, the minority of organisms that are present within the gut microbiome have already been sequenced and have a reference genome. Okay, so this is one of the hard problems. I'll come to that again in a moment. But part of the problem also is that taxonomy isn't the whole story. Okay, so for this, I'm going to ask you to participate again. This is a beautiful picture of E. coli. And I want you guys just to reflect on how you're feeling right now. So this is like me channeling my medical school psychiatry rotation. And when you look at this, how many of you feel good? Raise your hands. Okay, some people over in that corner and over in that corner. Okay, how many of you feel kind of bad? Okay, some people. And how many of you feel like, it's kind of complicated? More people. And the reason you feel like it's complicated is many of you who have ever worked in a biology lab, which is, I would presume to guess most of you, have worked with E. coli K12. Right? It is a workhorse organism of molecular biology laboratories. It is a rare member of our commensal microbiome. Usually doesn't cause us any problems. But you also know that there are virulent strains of Escherichia coli, like EHEC, that have caused terrible disease in outbreaks. So E. coli is not E. coli is not E. coli. And so while taxonomically they're the same, and that's what we can resolve using 16S sequencing, because remember 16S sequencing looks at evolution of the 16S gene through the acquisition of SNPs, we know that bacteria actually can evolve new functions and phenotypes through horizontal gene transfer or acquisition of new genes. And it is indeed this mechanism that provides lots of organisms capability for virulence. Okay, so taxonomy is not enough to infer function or clinical relevance. I touched on the fact that your microbiomes are filled with microbes that we haven't sequenced yet, that aren't in any reference database. How do we actually come up with references for the near 1,000 bacteria that are in your gut? Well, we can try to do so de novo. We can collect your stool, extract your DNA, sequence in little tiny DNA fragments. Then we can try to stitch things together. We can look for areas of overlap between the DNA fragments and daisy chain them together into longer and longer sequences. That's actually what we've done in this experiment here. Okay, so this is stool from a patient who had graft versus host disease versus stool from an individual who had no colitis, no diarrhea after transplantation. Okay. What we've done here is every single long piece of DNA that we were able to stitch together, we represent as a circle on this graph. We've only represented contigs, which are these long pieces of DNA that are longer than 2.5 kb. So already pretty impressive that we could go from 100 base pair segments to long contigs. We then array them based on %GC content, which tends to be conserved for any given organism if you look over a long enough period of sequence, as well as coverage, which is another statistic that we would expect to be conserved for contigs from a given organism. And what you start to see here is little clouds of circles that go together, which are proposed draft genomes. So already, without knowing anything about the taxonomy of this community, or these two communities, you know that the one on the left is far less diverse than the one on the right. But the problem is that you really can't figure out how these circles are connected to one another, right? They're not like one big, nice circular chromosome. They're genomes or suggested genomes in lots of little fragments. And so, we don't have a reference genome for most human-associated microorganisms. And going from these metagenomes, or draft genomes, proposed genomes, to actual genomes has been hard to impossible. Okay, so why is it hard to impossible? To describe that, I need to give you a little toy example here. Okay, so these are two genomes, two linear genomes, ARD, the other one is BRC. In the middle, you can see that that repeated segment R is the same in both organisms. Okay, so when you do these sorts of short read assemblies, this is what your assembly spits out at you. It says, well, A and B and C and D are all contiguous, but I'm not sure what to do with R, because it's present in two different places. Put another way, this is what the graph problem looks like. This is actually what the assembly graph looks like. And the problem is you don't know how to trace the proper path through R. Now let's say the proper path through R in this case was BRC. Well, one way you could trace the path would be to just generate a super long sequence. And it turns out there are methods to develop super long sequence. And so we thought, great, we're going to try long read sequencing, like PacBio sequencing or Oxford Nanopore MinION sequencing. But it turns out that this is very difficult, in part because the mass input that you require for these sorts of approaches is very high. And so unfortunately, while we would have really liked to have long read sequencing, we weren't able to get it to work. And so what we wanted to do was find another way to trace the path through these repeated sequences to try to get more contiguous genomes. So resolving this sort of structural variation or placing repeated sequences is hard. So this is where we decided to adapt a new technology. And it turns out that because the microbiome area of research, though up and coming, is not a major target for technology developers, one of the things that we can really benefit from is finding analogous situations in, for example, human genomics or cancer genomics, where there's a ton of technology development. And so what we decided to do was actually to find an analogous problem. And an analogous problem that happens in human genome sequencing is trying to do phasing or haplotype phasing. Okay, so the idea of haplotype phasing is if you have two single nucleotide polymorphisms that are present on a single copy of the chromosome, let's say it's your maternal chromosome, and they're really far apart from one another. Short read sequencing won't allow you to figure out whether they're in cis, so on the same chromosome, or in trans, one on the mom's chromosome, one on the dad's chromosome. And so a company called 10x Genomics had created this platform to essentially be able to trace paths through long human genome sequences to try to phase those variants. Okay. Basically what this does is it is an oil immersion technology. It uses a microfluidic sort of chamber to come up with single long pieces of DNA trapped within a bead from which you can generate sequence data. Okay. Put another way, the way we applied it was to take a mixture of microorganisms, extract DNA, isolate the high molecular weight DNA, and then in each and every one of these beads, and we can generate 1 million beads at once, we have a single piece of DNA that's trapped, and every single sequence that comes off of that long piece of DNA will be tagged with a specific barcode. Okay, and in this particular case, we're going to tag it with a red barcode. So this was a commercially available technology. Unfortunately, there was no software available for the metagenomic assembly application, which is a bit of a difficult situation. And so that's actually what we spend a lot of our time trying to do, rejiggering this technology to work on our particular use case and coming up with new software. Okay. Put more simply, what we decided to do was use this technology to be able to trace a path with those specifically barcoded reads through the right pathway on this graph. Okay, so how did it work? One way we can test how well this works is to actually test it on a synthetic mixture. So we created a synthetic mixture of 10 different microorganisms. We know that there's actually a great use case for this in bacterial assemblies because the 16S ribosomal RNA operon is actually very conserved between bacteria. There are different mutations in it, which allow us to do taxonomic classification, but this is essentially a repeated sequence that's roughly shared between all of these organisms, which means you end up with a graph that looks like this, okay? A repeated segment in the middle, that's the 16S locus, lots of choices on the left side, lots of choices on the right side. And you might also remember that bacteria, many bacteria have more than one copy of this. So some bacteria have up to 10 copies or more of the 16S operon within the genome. When we did short read assembly alone, we were only able to assemble one collapsed repeat. When we applied our new method, we were able to actually generate many more of these collapsed these repeats in the proper position. And we confirmed this using Sanger sequencing. And so to make a long story short, we can apply this method to get more contiguous genomes from a metagenomic mixture. And then we can also place these repeated segments in the proper context. All right. So remember, the whole reason we went on this technological odyssey to try to be able to resolve this issue was because we had this question, which is how does oligodomination occur? And so what we decided to do was to apply it on that individual. Okay, so you may recall the patient who had those 4 time points, A, B, C, and D, and that one organism in the light blue box ended up dominating. That was Bacteroides cacchi. You know that that organism was present at all 4 time points, but dominated between time points B and C. This is actually the de novo genome of this organism. And this is about the contiguity of the genome. This is 1/6 of the genome that we can get for the different time points using just the traditional short read approaches. Okay, so you can see there are lots of holes. When we applied our assembly method, the Athena assembly method, we ended up with much more contiguous sequence. What this allowed us to do was something interesting. And this is actually one of the things that we were able to show. So if you look at that long blue bar, that is the assembly that we have from time point D from Bacteroides cacchi. You can see it's completely contiguous. Below it, you can see what the short read assembly looks like, lots of holes. When you take a look at what's in that long 60 kb fragment, it's actually a horizontally transferred gene segment. It has a bunch of antibiotic production genes, et cetera. Okay, so this is an example where in time point C, that segment was absent, and in time point D, that segment is present, which means what you are seeing here is active horizontal gene transfer between time point C and time point D. This is how bacteria actually acquire phenotypes like resistance. So I think this is really exciting because we've actually genetically seen horizontal gene transfer happening in a single patient by resolving metagenomic data. What else can we do with this? This is another example of something we were able to resolve. Insertion sequences are kind of minimal horizontally transferred or internally transferred sequences. They encode transposases, which are genes that cut and paste DNA, okay? Or they can copy and paste DNA into different places if they're retrotransposons. Okay, but this is a transposon. It cuts itself out. It can paste itself in somewhere else. And it's present in about 30 copies throughout the genome. Okay, this is a perfectly repeated sequence, so this is the sort of thing that really breaks assemblies traditionally. What we were able to do was to place these in the right context. And this is an example where at time points A, B, and C, where you see those filled circles, the insertion sequence was present, but at time point D, it went away. Okay, so the open circle indicates the insertion sequence left. We can confirm the presence of these insertion sequences by generating primers to the flanks of where we predict them to be. And if the insertion sequence is present, you'd expect a higher molecular weight band. And if the insertion sequence is absent, you'd get a lower band. Okay, so these are 4 time points, A, B, C, and D. You can see the insertion sequence is absent at time points A and B and D, but it actually pops in for a little time at time point C. Okay, so you're actually seeing a shape-shifting genome over time. Here's another example of that sort of transposition event. This follows a bit of a different pattern. At time points A and B, the insertion sequence is absent. And at time point C and D, it's basically present and fixed. Let's examine this a little more closely. Okay, so this is the genetic neighborhood of that particular insertion sequence. The insertion sequence has the little pink box above it. You can see that the gene runs in this direction. Okay. Interestingly, this insertion sequence also has a fascinating feature, which is that it has a promoter, which is indicated with an arrow, that can drive transcription of a nearby gene. Okay, so it just carries around this bioregulatory element that'll turn on whatever's to the left of it. What's to the left of it? That's NORM. Okay, this is a multi-drug resistance gene. It encodes a multi-drug efflux pump. Okay, and you would expect that if it was turned on, it would bequeath its organism with drug resistance. Remember, this gene, this insertion sequence, popped in at this location between time points B and C. So here you can see time points B, C, and D that are indicated with no insertion, insertion, insertion. And when we did RNA-seq to look at what was being transcribed, you see exactly what we would predict. Okay? In time point B, you have basal transcription of this antibiotic resistance gene. In time points C and D, the transcription jumps up dramatically. Okay? This is a situation where no gene came or went, but this transfer event actually ended up driving expression of a gene that conferred resistance in this case. So what have I talked to you about? We talked about some bad diseases like leukemia and lymphoma and some risky treatments like hematopoietic stem cell transplantation. We also discussed how many of our patients end up dying of infection and GVHD, and we need to address treatment-related mortality. We talked about a common phenomenon that's associated with this treatment-related mortality of oligodomination, which has been associated with poor survival. We asked one simple question, which is how does oligodomination occur? We tried to solve 2 hard problems. One is inadequate reference genomes. The other one was difficult to resolve structural variation. We applied a new technology, that's read cloud or linked read sequencing and the Athena assembler to try to solve this problem. And we were able to identify some intriguing results, results that suggest that these organisms are alive and changing. They are actively adapting to perturbations that we provide. And you can see genes coming in and out. You can see active horizontal gene transfer, and so on and so forth. So with that, we've kind of been through these conclusions. I will close by telling you that one of the things that we really need to do in the space of microbiome-directed therapeutics is figuring out how we can manipulate the microbiome to improve outcomes. Our laboratory is very focused on trying to address cancer and chemotherapy-associated diversity loss. We've been trying to do this in 2 different ways. One, by practicing antibiotic stewardship and figuring out what antibiotic stewardship should look like in this very, very vulnerable population. And on the far right, we've been doing clinical studies in patients with prebiotics or microbially accessible carbohydrates that selectively feed elements of the microbiome. We've already completed a phase 1 study. We're moving to a phase 2 study next. So with that, I will thank my lab, our many patients who participate in this study, and my funding institutions. Thank you.